All right, guys, this is another episode of Harry Matters. This is episode 14. We have our second repeat guest ever, Professor Tais Gratieri, and the last time we had her on was about a year ago when we were much earlier on in this topical dutasteride study that we've been running now for about 2 years. We're coming up on the 2-year mark. And so with that said, thanks so much for coming on, Tais.
Well, thank you very much for having me again. I really love the name of that podcast, by the way.
I wish we could take credit for it. I'll give credit where credit is due. That is a Ralph Paus special. And so yeah, but I mean, I think a good place to start could just be really for the audience that maybe is less familiar with the work that we've been doing now for the past couple of years. I think just a really high level summary of kind of how our relationship with you started and then the work that we've done over the past 2 years.
Well, our relationship, we have a funny story behind it that you don't know of. And because you guys sent an email like, whoa, okay, we have seen your work, your scientific work, and we have seen you're like an expert on hair follicle delivery, and we would like to fund the research. That was the first email you guys sent me and Professor Guilherme. So I read that and I thought, that's a scam. And I deleted the first email because I thought no one knocked at your door and said, here, I want to fund you. That doesn't happen. So I thought that's a scam. And I deleted the email. But then Professor Guilherme had more faith in humanity. And he digged to look, what is this Tao thing? And what is this doing to science? And he was doing some research and thought, oh, that's a real thing. Communities are funding research. That's a real thing. Let's talk to them. And I said, yeah, really? And then we scheduled a first interview. So we were like here locked in our labs, unaware of this movement. So that was really interesting. And that has changed a lot the way I think and the way I think now science is progressing. Because if we look back at the work we have done in this past 2 years, in normal grants, that would have been impossible. I mean, that's sure to say that we have delivered like 3 times more of the work plan. Like when you say, oh, please deliver me a scientific work plan for 2 years research. And then I had, well, okay, I have one person working in this, and then we can do this, this, and that. And then we have seen like the way things work, and it's not like one person developing a grant and having all that time and the word. It's like a team fully devoted in solving one problem. And then it was problematic at the beginning. Dutasteride is a very challenging molecule to work with. So we began with one person, and then we have like 5 fully committed to solving this. And then things were progressing really fast. And in these 2 years, we have done a lot more than we even expected. So it has been really nice.
No, totally. And I'm curious. So are you saying just like the normal grant process, it's more of an individual thing, or it's sort of like you can plan for sort of less time in the future, or what's the difference, I guess, you've really experienced there?
Yeah, I believe so. Because, well, if I have like one grant and then I select, here we're the new university. So we select a student for a master's student or a PhD student to develop that project. And normally it's one person. I mean, we have a team that is working together, but with this, we could find like postdocs. And that's not so common to have. Like we had a team with 3 postdocs fully committed to this project. So things progress much faster. It was not someone that was learning how to use the techniques because, well, of course, we had PhDs, we had master students helping, but not fully in charge. And when you have this grant, well, we have to teach them how to teach a technique. And there's this learning time. And with this, we had the postdocs that were already very experienced in all the techniques we were working with. So things were much faster. And yeah, every time we had a problem We planned for like one postdoc at the beginning, and then in the end, it's like, oh, that's a complicated molecule. Let's get more people involved. And so we had 3 postdocs fully committed.
Yeah, I mean, we were super impressed by how much, just how many vehicles you tested. I mean, I think the final total, you correct me if I'm wrong, but we were over 60, I believe.
Over 60 vehicles, and 40 of them we really tested in skin's in vitro experiments, 2 time points.
And like we said, right, this is the reason this is quite a unique study, is that even if people have, certain like penetration data or drug targeting data for dutasteride, it's all been in different models with different methodologies, with different measurements. So there's never really been a broad-based apples to apples comparison of, specifically dutasteride delivery.
Yes, it happens with dutasteride and also finasteride. If you look at the papers and the literature, you can say, but there are particles described here to deliver finasteride or dutasteride, and PLGA nanoparticles and nano emulsions and all these types of formulations. But one have compared in human skin ex vivo, and another one has compared in mouse skin. And another one has used full thickness skin, and the models are different. So one has only, what's normally is done is people often treated this as transdermal. So transdermal means penetrating through the skin, and they're collecting the samples from the receptor compartment, like the fluid beneath the skin. And then you can say these formulations delivering more or less, and comparing what has been through the skin. And many of these papers are not looking into the hair follicles. And that's what we need to get into if we're looking to treat alopecia and something related to hair follicles, that we want a topical delivery and we don't want side effects. So we want the drug as maximum as possible within the hair follicle and not penetrating through the skin or within the skin, because follicles, that's a pathway of delivery. So the drug gets it into the follicle and then it diffuses from the hair follicle to the rest of the skin. And that's something that happens with every drug. And it happens a lot with the dutasteride. So you want a formulation that can hold as maximum as possible of the drug and slowly release within the hair follicle. So that the small amount that is being released finds the receptors there and have their actions there in that environment, not go to the other layers of the skin and eventually comes into the systemic circulation. So that's very challenging.
That's why we're of the opinion that delivery is just so important, particularly when it comes to hair loss, because what you currently have on the market, even with a lot of normal high dose topical finasterides, the problem there is typically not that it doesn't get through the skin. Like with dutasteride, sometimes you have issue actually penetrating the stratum corneum, but with finasteride, it's like they just dose such a high dose of the topical, and it all just sort of indiscriminately goes through the skin and gets absorbed into the viable skin, all the vasculature that's there, to the point where you have so much finasteride in your bloodstream, you might as well have just taken the pill and all the side effects. But yeah.
Yeah. And that's the perspective of the industry, because, the drug is approved for oral use. So, okay, so it's like, okay, penetrated a little bit, that's not a safety issue. But it will trigger side effects and that you don't want.
Okay, so Tais, I think a good thing to just give, to tell people a little bit of an overview. So obviously we were testing, dutasteride delivery, specificity to the hair follicle across over 60 different vehicles. So how exactly did we do that? What was the model used, in your own words?
Well, we begin with developing particles to target the hair follicle because, the drug is small and it's very lipophilic. So one thing would be to put it in oil. So it's soluble in oil and so on. But that's not a viable option because then your head would be all oily and stuff. So if we put particles, we begin with different types of nanoparticles. We say nano. People tend to think that it's something very small. But when you compare to the drug molecule, nano, it's like something very big. It's way bigger than the molecule itself. So in the skin, well, you have the stratum corneum that protects the skin. It's a layer of dead cells and lipid content that protects the skin from losing water, basically. So it's a very good barrier. But you have the hair follicles, which are invaginations of the skin, and then the stratum corneum, which protects the skin. It also invaginates and it creates like this hole. Then the follicle comes, and then it's a pathway also for drug delivery, but for these nanoparticles to accumulate in these follicle structures. Because if you, for drug delivery, you consider it not so much of a relevant because of the surface area. Then if you see the surface area of the hair follicles, okay, in the head you have more, but compared to the rest of the surface area, so it's not so relevant for drugs because they're small and they interact with the stratum corneum and so on. But for nanoparticles, they're very big. And then when they come in contact with the skin, they tend already to accumulate in these invaginations and to be accumulated in the hair follicle. That's a natural tendency of nanoparticles. So the thing is, okay, so we thought about all the types of nanoparticles we could develop. And we had to think of these nanoparticles also having a lipidic nature to combine with the dutasteride that is very lipophilic. So a lot of drug would be combined in the particle. And another thing then is if the particle is accumulating in the hair follicle, then it should hold the drug because the molecules goes from the most concentrated to the less concentrating, following a diffusion gradient. So if naturally after it's in the particle and the particle begins to interact with the skin, the drug is released. And this is called drug release, actually. And we found for the dutasteride, the relationship is not always like linear. As much as the particle controls the release, as much as it's targeting the hair follicle, depends on the molecule. But for the dutasteride, that was very true. And then the particles that could control better the drug release were the particles and the nanosystems that were targeting the hair follicle. But that's not just it. It's target the hair follicle and maintain the drug in this environment. Because that's why we made all the experiments. We had 2 time points. We had 12 hours and then 24 hours experiment. Because then we had 12 hours, we evaluate what's the amount of drug and particles in the hair follicle. And then, well, what happens with time, with prolonged times? The drug is released from the particle and is in all directions to the other skin layers. And then when we compare with the 24 hours, we already see which particles maintained this targeting effect for at least 24 hours. And for the dutasteride that is very potent and the dose is very small, we had to do this very long time points to have enough to quantify in the analytical equipments. But for other more hydrophilic drugs or less potent, sometimes we can even test this in 6 hours, 12 hours, and so on. The time points vary. But for the dutasteride, that model was
and I think the
okay.
interesting thing to touch on too is sort of the fact that we used pig skin versus human skin, and then also maybe just for the people as well, it could be interesting to hear sort of how we quantified where the dutasteride was, whether it was in the actual follicular shaft or the viable skin or the epidermis, and then kind of you're the main, I feel like the main number that you really looked at to evaluate these different vehicles was targeting factor. So maybe explaining like what targeting factor means and the importance of that as it relates to drug delivery.
Yeah, sure. That's something we debated a lot at the beginning of the study, and I was defending the pigskin. The pigskin we use, it comes from the slaughterhouse. So the pig that was the meat that was going to be used for consumption. So there's no ethical issues there. But we need to collect the skin, and we collect the ears of the pig, And we need to collect them before some scalding process, because they have the sanitary scalding process in the hot water and so on. So we have to collect them. It's not something we buy in the market. It's like the skin freshly obtained, and then we have these special treatment to transfer directly to the lab and so on. Okay. So what's the difference between the piggy skin and the human skin? The hair follicle of the pig skin is a little bit larger. So you think, well, then it's not a good model. And that's the point, because, well, the human skin, we can have human skin from plastic surgery mainly, and abdominoplastic surgery, and the main source of human skin for experimentation is abdominoplasty. Then you have a huge size of skin, and you can use. But, well, in the belly, at first, you don't have as many hair follicles as you would have in the head. So to have scalp skin, that would be more complicated. But again, when you have human skin and you remove it from the body, what happens? Well, the skin is very elastic. It's covering all my body. So, but if I cut a piece and take it out, it shrinks. And then this shrinkage has been demonstrated scientifically that it almost closes the hair follicles. So the human skin is not a good model to study follicular delivery in humans. And the porcine skin, that happens as well. But as the follicle was larger, when it happens on this kind of shrinkage, then it's the same size and the same structure as the human skin. So it's very comparable to the human skin. And then that's a very good model. We can obtain it fresh, and we can use full thickness. That's something also very important. When we say full thickness, is that we just remove the skin with the scalp, and then we have the whole dermis. We don't like cut the dermis in half. So we have the whole dermis, and then the whole follicular shaft is preserved, all the structures. And then, yeah. Yeah, and then it's a very specific quantification methods. We use HPLC, which is high pressure liquid chromatography, and the method is validated to detect nanograms per milliliters of dutasteride. And then, well, we apply the skin in this setup, fully validated, again, controlled area, controlled temperature, everything. We use the skin, and then we apply the formulation on top of it and leave for 12 hours or 24 hours, depending on protocol. And after this, we remove the skin, we clean, and then we remove the stratum corneum with tapes, with successive tapes. We can use from 10 to 30 tapes, depending on the protocol. And then these stratum corneum removed with the tapes is also, we put in some organic solvent, remove this stratum corneum residue, and quantify the amount of dutasteride in stratum corneum. And then we use some cyanoacrylate glue and apply to the skin and remove all the hair follicles with this. It's called differential tape stripping. And then this material we also extract with organic solvent, and then quantify the amount of dutasteride in these all these hair follicles together. And the rest of the skin is also processed, and the drug is extracted as well. So this targeting is the relationship of how much is in the follicle compared to everything else. So it's like the percentage of the drug. If you look at the percentage of the drug that penetrated your skin, what's the percentage in the hair follicles? So that's the targeting factor. So these targeting factors from 0 to 1. And for example, we had very good results with the proliponel, these instant forming liposomes, which had, after 12 hours, 0.64 of targeting factors. It means 64% of the dutasteride penetrated was in the follicle. And even more remarkable is that after 24 hours, this value almost didn't drop. We had 58% after 24 hours. So it was holding the dutasteride and slowly releasing. So that was really remarkable. And for all the other formulations, in average, we had like 30%. That's normal to have like 30% of the drug within the hair follicle. And as much as you have above that, that's already very good. And ideally, you have over 50%. That's what we achieved. And also with some other polymeric nanocapsules, we could have high targeting of almost 50%. So we had like two options.
I mean, even with a lot of the normal liposomes, which I feel like a lot of other topicals on the market use as sort of just standard liposomes, I feel like we were seeing like 0.15 targeting factors, which I was kind of very surprised by how poorly they performed when that seems to be kind of an industry standard.
Because what happens is some lipidic particles, they interacted very well with the stratum corneum. So these very good interaction with stratum corneum, which is very good for many local, many topical therapies and even cosmetics, if you think. So the particle is interacting with the stratum corneum and already releasing the drug. So it was not holding enough to target the hair follicle. They were already releasing to the skin. So when you look at the percentages, so much more is going to the stratum corneum and the upper epidermis. And then you have lower percentages at the hair follicles, which in this case were the target for our therapy.
When you see a targeting factor of 0.6, what are the next thoughts that go into your head when you're determining a dose of like what the optimal dose would be given that targeting factor?
That depends on the clinical effect, actually, because then it's very hard to relate. I mean, we have to have these standardized in vitro experiments to compare formulations. So you can compare in vitro with in vitro. Like to do this in vitro in vivo relationship of those to estimate the drug that would actually be in vivo, that cannot be done in vitro. That we could have to test, like we could go to animal models to evaluate the efficacy and to compare how much, use the formulation and to compare the efficacy, and then you can have this dose related response. That for that you have to go in vivo.
We the first people who try this product, we should have them get some DHT tests and see how much is going systemic.
I understand it could be in compounding pharmacy because, again, in safety terms, it's very potent, but you can use a very low dosage, and it's approved to be used orally. I mean, you know the effects. It's being used off-label orally, and so you won't have a side effect as high as the oral dose if you're not using as high as an oral dose. So that's for sure. So
I think we used 0.09 milligrams per milliliter, if I recall correctly, right? With the thinking being that this topical, we could use a lower dose because more of it will get to the hair follicle. 0.25. Okay. Yeah.
0.25.
Gotcha. Sorry, Bex, do you have a follow-up?
No.
So when the drug is getting into the stratum corneum, the viable skin, or the hair follicle, the amount that gets stuck in the stratum corneum, that does not go systemic, right? It's only the amount that gets through the stratum corneum into the viable skin that goes systemic.
It depends. Depending on the therapy and depending on the drug, the amount that is trapped within the stratum corneum will eventually penetrate. So that's a matter of time because, well, you can have some very strong interactions in some cases that then it won't penetrate because it's having a strong binding to some protein or some lipid in the stratum corneum. And then, well, it's a dead layer that will peel off. And then when it peels off, the drug goes away. But if not, if you don't have these very strong interactions, well, you have a higher concentration here, and then it continues to diffuse to the other skin layers, and it can eventually go to the systemic circulation. That's why you want to target as much as possible in the hair follicle, because, okay, if you have some amount in the hair follicle, like 15% we had with conventional formulations. And then you have 85% in the stratum corneum. Eventually, this 85 will go somewhere. It won't disappear. It won't be metabolized many cases. You don't have the enzymes there to metabolize. So it continues to diffuse through the skin. And so that's concern. That's why it's very straightforward to do this. You go from 60 formulations, and then you have 2 that is outperforming, and then let's go in vivo. That could be a next step. Let's compare these 2, and then you don't lose time and resources. Like comparing 60, imagine how would that be? We would go by luck. And these in vitro experiments, and as much as control as you have, it saves you time in the end, and it saves you resources.
Yeah, I heard Verbinen's going to be the first person to try it. So
we'll be it'll be available in all 50 states soon. So why not? I want more hair. So I think something also that's interesting, Tais, that a lot of people ask, and I think we've talked about this several times, but they think, okay, so we found this amazing delivery vehicle for dutasteride that seems to work much better than all the other ones we've tested. Can we just use this with every other drug? And as we've discussed, obviously, it doesn't work like that. So I guess maybe speaking to that a little bit, but then also sort of more specifically what you found difficult, challenging, and maybe surprising about working with dutasteride.
Well, one thing, to begin with, the difficulty is it's very potent, so the dose is very low, and we have to extend time periods because, we have tested many time points before deciding to work on this 12 and 24 hours. You have this time for validating the experiment and the time points and the extraction methods, because normally it's much easier to extract like from the stratum corneum and from the hair follicle and from the skin. I said we can extract with organic solvent. So if you put 15 ml, 10 ml, a lot of organic solvent, it's easier to extract. But we couldn't dissolve the drug. There was very few, and so we could detect it in the analytical procedure. So there was a challenge to adapt all the methods. And then another thing is it's very lipophilic. And the conventional formulations were having this behavior of interacting well with the skin, and it was interacting well with the skin, and a lot was accumulating in the stratum corneum and viable skin, which is what we didn't want. And then was challenging to find something that would really outperform. And but it won't work the same way for other drugs, and even if you think like, okay, but you have now artificial intelligence, couldn't we predict all these interactions? I mean, that's something that researchers are trying to do a lot for the past years, and it's very, very complicated. And we still don't have a model to predict permeation. And we look at, of course, we look at some parameters of the molecule, like the size of the molecule, the lipophilicity, and so on. But you can still have some unpredicted interactions with compounds, with molecules and proteins in the skin, because it's so complex. The biological matrix is so complex. It's very hard to use all these variables and have these complete predictions. And when you add the formulation factor to it, that's another complicator, because the formulation will completely change the behavior of the penetration. And then it's right now at the moment, sometimes maybe in 10 years, we'll have something to predict this. But as the moment we are now, it's faster to just run the experiment than to try to model all these things. It's straight faster to just do this experiment and
it's our perspective that a lot of these models, in order to really be predictive in the first place, they need more data, apples to apples comparison of how delivery vehicles work on the same models. They need more drugs tested on human hair follicles before they can really have more predictive power. So we're totally aligned on that. Real quick, you mentioned a couple of the variables, like size of the vehicles, charge of the vehicles, which I don't think I don't think you met. Yeah, the polydisparity index,
Charge, of course.
lipophilicity.
Yeah.
Are there any others that you kind of look for when you're thinking about delivery?
Well, yeah. Well, you look at the charge at pK, pKa, pi, depending on the molecule, and lipophilicity, size, and that's it. And some molecules have some strong interactions with others. Some form aggregates, and then you have all these behaviors that can happen.
We found what we're really excited about with the Pro Lipo. I think, were you overall pretty satisfied with that? Like going through this whole experiment, were you pretty excited to find Pro Lipo?
Yes, indeed, because that was something ready and something, again, that's something fast that was available already. And well, the thing is, we had this multi-layer. They say it's multi-layer, multi-bilayer. So one layer, another layer, and that ends up protecting the drug. And well, there are other. We found a brand that was lucky enough to have these extreme, very good targeting. But there are other brands as well with this instant forming liposome. So yeah, there would be more to test if we wanted to test others, and also polymeric. And then again, the goal is like to go nailing down to the best ones. And this was the one that best performed so far, and it's easy and it has it's markedly available.
So I guess once we found this and we were super excited by the targeting factor, now it might just be cool to talk a little bit about sort of the follow-up studies that you did, which I think are most relevant to the topical actually being used in practice. So the sort of stability testing you just did, the irritation data you collected, and then actually the release data as well. So I don't know if you want to just speak a bit to those three, because that's particularly relevant to somebody if they're gonna have the drug vial in their cabinet. And
Yeah, that's something that is different in the model we use to work with the hair dye, because we wanted to have what's the more relevant results, and then we were progressing, testing all these formulations, and then we went back to like compatibility issues, evaluating all these experiments with the compatibility between the drug and the excipients of the formulation, and then to evaluate the drug release, because we thought, well, again, it's faster. Let's just see what performs well in the skin, and if it's good, then we'll complete the experiments with the drug release, and to have all the controls, and to state that there was all these steps were done, but we did it in the inverse order. But then we completed with and also the stability. So we maintained the formulation in 3 temperatures for 90 days. And then we were evaluating every time point, like the pH, the size of the liposomes, the drug dose that was within the liposomes. So the amount of drug, the drug was not degrading. And then we also evaluated the sensitization with if to see the formulation would not be irritant to the tissue. And we used also an in vitro model. It's called the HET-CAM test. It used embryonized eggs, and then you have like the eggs after 10 days of fertilization. You just expose the chorioallantoid membrane, and then you evaluate if the formulation is not irritating, if not provoking some hemorrhage or redness or something like this. And you compare. It's also a comparative test. You have some negative control, positive control, and then you observe, you use your formulation and you observed it for 5 minutes. And then there's these scores, you tested and so on. And all these formulations were like completely non-irritant.
yeah, that was super exciting for me, because I know everybody's always like, I tried this, I tried that, and my scalp got all red and irritated. And we should totally put up the picture of the membranes, because it is kind of cool to look at. You can see like visibly in the irritating formulas that you did use sort of as your controls. You can literally see it's like a rash almost, or I don't know exactly. I mean, it looks like irritated skin essentially, but
That's a big deal with the formulations containing minoxidil and all the formulations that are more lipophilic, that you need something to increase the amount in the formulation. So you use like propylene glycol or some penetration enhancer, and these all work well in the first week, but then you start to get irritation of the skin, and then your conditions worsen. So
and then
that's
just the
something not
stability as well. I think you
admittable.
tested over a 90-day period stability, and I think just the maybe touching on the variables that you looked at could be interesting. What
Yeah, we looked at the fridge environment, room temperature, and 40 degrees. And then after 60 days, at 40 degrees, you start to see some changes in pH and in drug amount. So it indicates like the stability will be um, lower if you not preserve it in very high temperatures. But as the stability in fridge was okay, so if you keep it in the fridge, that would be recommended. And it's not so much of a problem for you guys there in New York and US, but well, here in Brazil, we can have 40 degrees room temperature, so that would not be good.
So I had a couple community questions before I went it, and then we'll go into sort of future research, potential next steps. But Bax, was there anything you wanted to ask? Maybe you wanted to ask one of the community questions that are
From Salvation, are the existing topical dutasteride vehicles any good? And referring to the ones already sold commercially, or do you think most of them fail because of poor delivery?
Well, it depends. We have seen that these traditional formulations, like the ones that are formulated like minoxidil and so on, they had around 15% of follicular targeting. So the ones we tested were not performing very well, but well, I can say I can reply from the ones I haven't tested.
a lot of these. I will caveat a lot of these questions we need to ask you because they'll get angry with us if we don't. But we understand that they're very difficult to answer, and so at least they'll believe it coming from you, at least that it's impossible to know a lot of these things.
So there's three. So the next one from Kefir, formerly known as cat belly rubs. Since there are vehicles that can carry both lipophilic and lipophobic APIs, APIs, for those that don't know, just refers to active pharmaceutical ingredient, also just the drug. Do you think it's possible to develop a more universal delivery platform that could handle up to 80% of actives out there?
We have some depends on what you call like universal platform. Like for dutasteride, this proliponel is a multi-instant forming liposome, worked very well, and it could also encapsulate some more hydrophilic drugs as finasteride. So it could work well again with finasteride, but we would have to test them to be sure. Again, to have the encapsulation and to have the formulation, it's not something so okay, it's challenging, but it's not the end of the world. You run some tests, you have some options already developed and already sold that can work. And well, we tested liposomes, transfersomes, etosomes, multilamellar instant forming liposomes, nanocapsules, where you have like this shell, this polymeric shell and oily inside, and you have polymeric particles that work well with hydrophilic and lipophilic drugs. So we have many options that work with many types of drugs, but to see how will these interact with the skin and how will the drug interact from the particle to the skin, that's something that depends for every molecule. It would be different for each molecule. So you can only know for sure when you do the tests and you compare with the controls.
Totally. Always have to validate. And so then the last community question from Romeo: if you had to estimate the systemic absorption of our topical dutasteride based on the preliminary data, what would it be? And what confidence would you give in that number?
Well, I cannot estimate. I cannot estimate with the in vitro experiment the amount. That's something that someone would rip my diploma if I said so. If I could do that, that's it. That's not possible. But we can have like some indications. And a very good indication we had at this in vitro experiment is that we could not detect dutasteride through the skin. So in this experimental setup, we did not detect dutasteride through the skin. That's something I can say for sure. And to estimate how much of the drug would go like in vivo, it would have to be in vivo and much in adding another variable that is repeated administrations. And then, well, we can have these designs could be done in clinical studies, but it also could be done in some animal models. Then you apply repeatedly after some time and you follow the amount of drug in plasma if you can detect or not. And then you can have some estimates. Through the skin, that's the first step. That's the first thing. Because if it was being detected in huge amounts through the skin, that would mean the chances are very high of having a transdermal
100%.
penetration.
And then,
So
but because the
it's
skin
already a
does
good
have
start.
blood vessels, and some amount is in there. So to run HPLC or LCMS on basically human serum from people that are using the drug, would that be sort of the best recommended test in your eyes to quantify how much is going systemic?
No, to analyze how much is going systemic, you would have to go in vivo and systemic. Because you see, to have the drug in the dermis, we had some amount. Okay, we had 64% in the hair follicle, but where were the other 36%? So it was in the stratum corneum, epidermis, and dermis. So we had some in dermis. But to have some drug in the dermis doesn't mean a systemic effect. Because when you have like a local delivery, you can have a local distribution. I mean, the drug will act when it finds enough of the receptors at the enough concentration to trigger an effect. So you have a more concentrated amount in the area where you're applying. Doesn't mean you will have it in the blood distributed to your whole body in enough concentrations to trigger an effect. Like if you have some muscle injury and you apply some cream and like anti-inflammatory cream, it goes until it reaches your muscle, and you have capillaries there. It will reach local circulation, but it doesn't mean it will penetrate the systemic. It will from these blood vessels be distributed to the whole body in enough concentrations to trigger effect in the whole body. It's more concentrated where you apply. So doesn't mean you need to have 0.0 dutasteride in the dermis not to have a systemic effect. It's not like that.
Thank you. Well, that's it from the community.
Cool. So yeah, no, I mean, look, hopefully by the time this episode comes out, this new topical dutasteride that we're super excited about from a targeting factor standpoint, from a stability standpoint, from a drug release standpoint, and actually from an irritation standpoint or a non-irritating standpoint, will hopefully be live on antigen.xyz in all 50 states, at which point people can try it. Fingers crossed on how long that takes. Now, this has been sort of the conclusion of a 2-year study that we've been running with you guys. And so I believe our contract ends at the end of 2025. But that said, you have at least three really compelling potential ideas for studies that we could run next in collaboration with your group. So maybe as a starting point, the sort of it could be really great and cool to hear the 3 things that you were pitching to me, and maybe even if there's a preference between the 3 ideas that you have, because of course there might be. I know there's one idea in particular that our community really likes, and there's a back and forth.
Yeah, there has been a back and forth. Well, science never stops. If you leave me, I would go on testing other things for the dutasteride until it gets better and better and better. but it's not like that. We have to move forward. But well, there are some things that we can do. I mean, dutasteride has one mechanism of action. Well, and then we have seen studies that that also relate antioxidant effects to better cell function, and that eventually stimulates hair growth, and ways as well to impede those ROS, that oxidation stress, to go to telogen phase. And so antioxidant seems to do have an effect, a protective effect in the hair follicle cycle. But, and you see all these cosmetics, and they're sold as cosmetics, as tonics, and you say, oh, but I have used that doesn't have a very good action. That's not enough. Okay, that's not enough. But what if you combine both? So again, you have something that has an effect, so you have something that has another effect, different, complementary. So why not complement all these things in one formulation? And I think that's the future for treating hair loss. You have multiple pathways in the same formulation, so you cover it all up. So one proposition was to study this combinatory, and we have been studying already combination of minoxidil with latanoprost, which is another drug that has shown to be very potent in inducing follicle production, hair follicle production. And it's already very challenging to work with, very lipophilic, very potent. But we have advanced that already to have a formulation that could combine both minoxidil, that is not so lipophilic, and latanoprost in the same formulation, and target the hair follicle. So that's something that if we could evaluate, but we still would need to evaluate more time points, drug release, and some effects in cells. And if we could continue to work on that, I believe that's something we would not come from zero, but be already very advanced. And I believe that would be good to see these combinations, and also with these antioxidants, because then, well, we have different things. We had some antioxidants that have shown to work, and they all have some problems, some stability problems, and like melatonin and quercetin, luteolin, and there are some antioxidants have been really also related to the graying of hair, like the color, changing the color. They protect these events of having the hair to be gray, to be white actually. And so why not combine? You want to preserve your hair and you want to preserve the color of your hair. I think that would be something very interesting to work with these combinations. That was one proposition we made, like to evaluate minoxidil, latanoprost, and another drug with different antioxidants. These other drug could be finasteride or dutasteride. And I understood that the community was also wanting to have some finasteride drug delivery vehicles. So that's something that also be done the same way we did with dutasteride, to test different vehicles for finasteride and see which one of them will target best. Of course, we could begin with the ones that work better with dutasteride, but I believe we would have to evaluate other strategies because the drugs are different. Another thing is to discover some new molecules and some things that might have an effect and might have an antioxidant effect, for example, but something that can work in the follicular metabolism. And we have here the Amazon, that is very rich in biodiversity, and we could even sample and test new materials, like to do these bioprospection, to evaluate a lot of molecules and see what has an effect, a good effect. And well, of course, that's a bit different than what we're doing so far, because so far we want to test this molecule, and we developed the drug delivery vehicle and for that molecule, and to find a new molecule with a new effect, then you would have to test in vivo again, and maybe in an animal model. So see in animal model, and we have some partnership with some researchers in Pará, which is in the Amazon, and they are developing animal model. It's still being validated, so it's early to say that that's established, but still being validated this month. That it applies testosterone to the mouse, and then it first shaves the hair follicle of the mouse, and then gives testosterone. So the mouse that is receiving testosterone, the hair doesn't grow up, and then you follow by the control that is not receiving testosterone, and the hair grows back again. And then you test your formulation in another arms of mouse that are receiving testosterone, and then you apply your formulation, and you see if your formulation can control these. He's receiving testosterone, so the hair is not growing, but how your formulation can treat it, and then you compare this if the hair is growing back with the formulation. It can be with the formulation or with the new active, for example, so you can test many things. That's something really excited I wanted to do about. And then I left the best for last, because I know what you guys really want to look at is using antisense nucleotides to see how it could modulate and have cell events related to cell repercussions related to maintaining the anagen phase and maintaining the hair growth. And I had to look at it and talk to some other researchers here and another group that is working with this, and that's really interesting indeed, because we can have these antisense nucleotides that interact with some RNA. So you have a gene that is producing lots of proteins having multiple effects in the cell, and then these genes are expressed, they're produced. You have an RNA in the cells, and you have these ASOs, which are nucleotide sequences, can be small, and they can interact with these RNAs and block the chain of producing these proteins you don't want, and or the inverse, or to stimulate the produce of something else. And there are some ASOs already evaluated to trigger the twist gene, and it has shown, and it has studies showing that these has repercussions with hair growth as well. So you have these sequences, and it has been studied for treating cancer and some types of cancer, but not these repercussions related to the hair growth, and that's something that could be done. So, and there are many more steps for this research. That's something different and more challenging because it's very new. And there are many steps because you need to first validate your protocols. Like you need to put this inside the cell. It's called transfection. So you have to have this transfection protocol that doesn't kill your cell, the cell you're studying. So this would be done with dermal papilla cells. And then you transfect this material, and then you evaluate. You have to evaluate the molecular levels, the repercussions in molecular levels, and quantify the amount of RNA and the effects and the genes expressed and so on. Well, you can evaluate the cells in terms of viability, migration, proliferation, senescence, and then there are many steps. So first you have to have all these controls and to validate the experiment, the protocols, to see what's the repercussions in molecular levels, then what's the repercussion in functional levels, and then to develop a formulation that could make these ASOs get inside the cells. Because for the in vitro experiments, you would have a very controlled transfection protocol. But then in vivo, you would need some formulation to maintain the stability of these ASOs and to have a good transfection efficiency. So that's something, again, to be evaluated in cell culture as well. And it's challenging, and it's a different way of working now because we wouldn't. The protocol is not like making so many formulations and see how much is in the follicle, but it's more than that. It's after it's in the follicle, if it will get your ASO inside the cell and to produce the effects.
Yeah. This would be follow up on the work that we've been doing at the Imperial College of London, testing our top gene target, Twist1, with Claire Higgins there. And then, obviously we're as we're wrapping up that work over there, we would be interested in, if we found something promising, how do we actually deliver it to the base of the hair follicles like we've been doing with this dutasteride. And then I think you mentioned pretty much the natural sort of organic ingredients that you would also be interested. It's the natural extracts and essential oils that you were interested in testing as well. And so I was just curious, on the mouse model, what would you just be planning? How exactly would you be planning to use the mouse model? I was a little bit unclear on that.
Well, I was planning of using it for the dutasteride formulations have already developed, to compare some of the best formulations we had, and also in the future for the next formulations and formulations combining different actives and different drugs. For example, it could be finasteride and melatonin, and dutasteride and melatonin, or minoxidil and latanoprost, all of these very well drugs, well known, and so on.
Wrapping up here towards the end, I think the last question, at least that I had, was if you had a preference of which of those studies you ran, which would you be most excited to pursue next?
I can say I have a preference, but I think there are some things that would deliver results faster. And as it happened with the dutasteride, if we just go through evaluating finasteride in different drug delivery vehicles, maybe in 1 year we would have finasteride formulation, optimal formulation. And to evaluate, to find a new antioxidant with an effect, that's something very interesting, but would take more time as well. I mean, that's just 2-year work plan and to have some data. And the thing is, if we develop a finasteride novel formulation and try to figure out what's the best finasteride formulation, for example, in parallel, we can continue to do this in vivo mouse model to test the dutasteride and finasteride formulations. And that's quick. And then for the next year, we could, next year would be the first year of the project. That's what I said. But then for the other year, we could start with the combinations. But again, if the ASOs is the preference of everyone, that's completely feasible as well. But we would need like this first year to have all these protocols very well established and all the controls, and to be sure what are the best ASOs to we would work with. And the same time the cells experiments progress and the protocols, then we progress with the formulations to evaluate the stability and the formulation. And the way we would target delivery here would be different. It's not by quantifying the amount in the skin, because that's not possible, because that's basic. We can extract like the ASOs from the skin undoubtedly and distinguish between the materials we have in the skin, but we can think of ways of binding some molecules, some fluorescent molecules, for example, to the formulation, and then have microscopy to evaluate how is the delivery and the preference. And that's something that would not be done for all the formulations and many formulations that was with this protocol. But then we would test like stability and the most promising formulations in terms of transfection efficiency. And then we would go to observe this within the skin and with confocal microscopy, something like that. So to different project and it would take more time to start seeing these results because, well, you have to be very sure with all the controls. And there are many controls. It's not just, okay, I validate this ASO, so let's do this. You have many variations, and you have to have these controls with small variations in sequence to be really sure that this effect you're observing it's a very specific interaction with the RNA and not some in a specific interaction that is producing some effect. And then you have to be very it's not a waste of time to invest in these controls and to have all the protocols very well established.
I did find that interesting when we were talking the other day, and I was like, so why can't we measure the ASOs in the skin? You're like, well, if you apply dutasteride and you see it's like a foreign substance, you would see the dutasteride in your body, but the ASOs are essentially indistinguishable from the various proteins and sequences that we would have in our bodies anyways. And so it's like there's, I found that was a silly question I asked at the time, but I found that interesting at the same time.
Yeah, but in this model with the skin and all these interference, that's But you could bind some foreign molecule to observe delivery and so on. I don't think it would be very produced because if we think of, okay, so bind a molecule and quantify the amount delivered, that's like a lot of work to have the in vitro test, to have some answers, and it's not worth it. So it's easier to just go to see the effect in the in vivo model, for example. So you have the formulation and you can see the formulation, mark the formulation, some microscopy, and then you go for in vivo. It's like because it depends on the way the project is designed, because it would be so much trouble to produce this in vitro. It makes no sense.
All right, that's all I had. Bax, did you have anything you wanted to say before we wrap up?
No, thank you for the great work, Tais, over almost 2 years now. It's awesome to see how much you guys have gotten done.
Yeah, and it's, I mean, I need to point out that it's also building like scientific material and products for hair doll as well. And I mean, we have published so far 5 scientific papers, and we have in these 3 months we're finishing the last 2, and so we're gathering the results and writing the discussion. So we'll have 2 more in the next months published, so it will be 7 papers. And the first work plan, I imagine, if everything goes well, we'll have 1 per year, so it will be 2 papers, and we have more than 3 times that. So we're very happy with this as well.
And yeah, I'll just be the first to say that, like, Tais, you've been incredible to work with. For anybody else who's looking into funding any research, she responds within like 30 seconds, nights, mornings, on the weekends. I think we had planned to test like 20 to 30 delivery vehicles, and she ended up doing over 60. Like she said, we were planning on like one or 2 papers, and it's turned into 7. So it's been really awesome working with you, and hopefully we continue this partnership. Thank you so much.
Thank you very much. If anyone wants to fund me, I won't delete the email, I promise.
All right. Well, that's been another great episode of Harry Matters. Thank you, guys.